My talk is a kind of historical talk about what has been done here in Politecnico during these 50 years of the laser at the Politecnico School. As the rector mentioned before, there is the first anniversary to be considered. November 29, 1863, and that's the time when the Istituto Tecnico Superiore, thereafter named the Politecnico, was established. So this year we are celebrating the 150 years of Politecnico. But from a laser point of view, we have another very important date that we celebrate 3 years ago, which is May 16, 1960, when the first laser action was achieved by Ted Myman at Hughes Laboratory. And this set the beginning of the big revolution, which has been the laser era. And then there is another date that we are celebrating today, namely February 1963, when the first laser action was achieved at the Politecnico School of Milano. And therefore, today we are celebrating the 50 years of the Politecnico, the laser at the Politecnico. So we are having this compound kind of celebrations. So the general summary of my talk is that I am making a few historical remarks on the evolution of the field. I will introduce our contribution within this evolution, and then, of course, I will reach my conclusions. The invention of the first laser. As I mentioned before, on May 16, 1960, the first laser action was achieved by Theodore Myman. And the scientific community was really shocked, astonished by this, because of the simplicity of the components used. The race to obtain the first laser started 2 years before, and if you want to read more about this, you can read this book. And in this race, there were many important institutions like Bell Labs, TRG, IBM, and so on. But everybody, the general consensus at this time was that if the laser had not been invented at that time, the laser was necessarily very difficult to make. But this proved to be not true, because Myman, with very simple component and very bright idea, succeeded in having a laser in a very simple way. So the simplicity of the component used. Well, the 3-level nature of the laser transition is somewhat difficult to explain, but suffices to say that very authoritative people of that time had declared openly that ruby would never work because it was a 3-level laser. And the type of laser excitation, namely flash lamp. So Myman, which at that time was a young student, was a young researcher, 35 years, won the race against these big giants like Bell Labs and IBM. And of course, people from the other side were not too happy about that. Here you see Myman holding the first lasers, and you see the components were really astonishingly simple. A flash lamp, helical flash lamp. In the center of this flash lamp, there is this small crystal of ruby. Ruby is corundum plus chromium plus sapphire, and then a container of aluminum. That was simple like that. Now, that suffices for the beginning. Actually, the activity that I started at Politecnico in 1963 came after a period of 18 months spent at Stanford University between 61 and 63. And I always advise people to go for a certain period of time of their life to the States or to any other advanced countries. At that time, Art Shallow, the Nobel Prize in '81, had just arrived from Bell Labs. Tony Sigman, another giant in the field, just moving from laser to laser research. And Dick Pantel, with whom I particularly worked, but I worked also with Sigmund and Shallow, was also moving from maser to laser research. And on that period, I want just simply to mention one of the pioneering experiments we made of photomixing on semiconductors. And I mentioned this paper for a curiosity. You may notice that it was volume first, the first volume of a new established journal, which now is very popular, namely Applied Physics Letters. And here is a particularly interesting picture in 1963 in Milano, the first laser laboratory. You may notice perhaps two aspects of this picture. The young face of the people involved from one side, and the absolute poverty of the instrumentation. You may notice wood tables and very primitive equipment like optical benches. Now I want to deal a little bit with one of the topics which have been quite popular. Avvertimi un momentino quando mancano, quando ho iniziato?
Ho iniziato 10 minuti fa.
Allora avvertimi, devo parlare 40 minuti, avvertimi 5-10 minuti. Solid state laser has been one of the topics which has been traditionally one of the strongest topics in the group from the beginning. Now, solid state laser means lasers in which the active medium is made by a solid. There are three periods that can be distinguished within this history of these solid state lasers, namely the first period, which goes between 1960 and 1965, in which many lasers were tried, starting with ruby. This is ruby, in which chromium substitutes aluminum at some site of the crystal. And then these were the second and third laser of the history, uranium on calcium fluoride and dysprosium calcium fluoride. And then in the same 5 years, all other rare earth materials like neodymium, erbium, ytterbium, and so on were tested. But by the end of this period, only 2 lasers survived because the others were not proved to be too much practically, namely ruby and neodymium lasers, which could be hosted in a crystal named YAG or simply in a glass. And then a new period happened after the 80, after 1980, which is usually recalled upon as the renaissance of solid-state lasers, in which the tunable solid-state lasers were introduced. And this was thanks to the fact that instead of exciting the solid-state laser by lamps, as it was done before, they were excited by semiconductor diode lasers. And of that period, in this field of solid-state laser, I'd like to mention, first of all, this seminal paper that we published in 1966, in which we were first able, for solid-state laser, to obtain behavior like theory would predict, namely to work in what is called single transfer and longitudinal mode. No one was able to do that before our paper, and I remember when I presented this work in San Francisco in 1966 at the International Conference on Quantum Electronics, people got really impressed by this work. And this established the beginning of solid-state laser working as they theoretically should do. And these are the pure modes in 66. This is what's called the TM000 mode. This is the TM03 and so on. And there are some stellar modes. And they were quite coherent. So they could produce quite strong interference fringes, a possibility which at that time was only possible with gas laser. Solid-state laser at that time were working not in a coherent way, essentially. They were not coherent beams. Now, as I told you before, after 1980, the tunable solid-state laser came in, and tunable solid-state laser comprise a class of lasers, a vast class of lasers, like terbium, this is wavelength, oscillation wavelength, erbium, which may oscillate at 2 different wavelengths, thulium, holmium, or, and I've signed it in red because that's an important contribution that we have been given to this field, terbium, erbium glass, which oscillates at this wavelength. And the same material can nowadays be put in optical fibers, silica optical fiber, that has contributed tremendously to the renaissance of solid-state lasers. A few words about this big, also important development of our group, erbium-terbium glass laser, in which this is a platelet of erbium-terbium glass, which is excited by a diode semiconductor laser at 976 nanometer, and you can do that in a system with a discrete element, but also in a microchip, very simple microchip. And this laser has got quite important development all around the world because it has reasonably high power for this application, high spectral purity, less than 1 kilohertz, single mode, tunable, and wavelength is completely safe for the eye, which is important for open-air optical communication because the application in which this laser has been pushed forward are for telemetry, metrology, and in particular open-air optical communication, point-to-point optical communication. And here the co-worker of this work, which is mostly contributing to this work. Now I move to another topic which has been historically very important in our group, namely biomedical diagnostic by short laser pulses. This is perhaps the first work we published. That was 1974 with Saki and Pren and myself. And I would like here to recognize other people who have been strongly contributing to this work. And this was at that time, was quite interesting instruments. We had a tunable system generating pulses of 1 nanosecond, which was sent through a microscope here, down to the spot of the microscope, so we could observe the fluorescence emitted by the specimen, in particular cells, chromosome, DNA, which could be then sent back to this sampling oscilloscope. So we did quite a lot of pioneering work with this system on acridine molecules intercalating DNA, or fluorescence studies of porphyrin, phthalocyan, and so on for tumor detection. And in particular, I like here to mention a particular experiment done by this group here. This is a surface tumor, and if you use a certain fluorescent dye which selectively attach to the tumor, and you observe the fluorescence under ordinary light, you don't see too much discrimination between the tumor and the surrounding. But if you do excite the same fluorescent material with short pulses, and you make an image in terms of time duration of the decay time of these pulses, then you get very strong enhancement of the image, so you localize very strongly and very precisely the limit of the tumor, which is also, very, as you know, very important for surgery, for surgical intervention. Now there is another topic which has been going on for several years in the same group, which has been quite important, namely the study of short laser pulses. When I mean short, I mean 100 picoseconds. Short laser pulses through diffusive media. In particular, when I talk diffusive media, I mean human body, for instance. Now, in a diffusive media, what happens, suppose you have this containing several diffusing scattering center, the red one, and several absorption center, the green one. Then what happens if you enter with a pulse? The pulse undergoes a kind of random walk through this scattering center. From time to time, it gets absorbed, and then that's the end of that photon. But in other cases, it gets transmitted, but meanwhile, all this results in a pulse output, which has nothing to do with an initial pulse. You start with 100 picosecond pulse, this log scale, 100 picosecond pulse, and then you get a pulse out, which may be as long as 5 nanoseconds, but the history of this scattering phenomenon contains information about all the scattering center and absorption center which are being traversed by the pulse, so that by adapting the observed measured data to the theoretical model, according to the diffusion theory, you can retrieve the absorption and scattering coefficient at the wavelength you are talking about. So now if you change the wavelength, you can observe different absorption. Or if you move at the same time the emitter, this usually is a fiber, the emitter, and at the same time the receiver, across and the other way, you can make an XY scanning of the sample, and so you can make an image. Or you can do that by scattering, back scattering. We will see later. If you do that, you can make a mammography, optical mammography, which has been developed by the group, in which you can detect the malignant lesion and discriminate against cysts, which of course are not malignant cysts. Because in one case you have absorption at the wavelength of the blood, hemoglobin. In the other case you have a reduced scattering because the cysts have less scattering than the normal tissue. And there is a collaboration, I understand a strong collaboration in a European project with Professor Lindberg, which is there. Or you can do another thing, getting back to the original picture. You can observe the light not being transmitted, but the light being retro-diffused at some point. And in this case, you can put several fibers plus several detecting fibers, and you can name an image of the region at the wavelength you are talking about, within the brain. And in this way, the volunteer here is performing a given task, like moving the hands. And then, if you look at the part of the brain which is known to be responsible for that movement, you surely observe that the oxyhemoglobin, the oxygenated hemoglobin, increases. This is indicative that the brain is working, and it means that it requires more oxygen to the brain. And in a time, with a time resolution of a few seconds, which cannot be done by nuclear magnetic resonance, because this is what is called functional imaging, studies of functional imaging, in which this technique is complementary, in a sense, to what you can do by nuclear magnetic resonance or by PET, positron electron tomography. So much about laser application in biology and medicine. Now we have another topic which has been characterizing our group for almost 50 years, namely ultra-short laser pulses. When I talk about ultra-short laser pulse, I mean pulses with duration from picoseconds, for a few picoseconds, to a few femtoseconds. Picoseconds means 10 to the minus 12 seconds. Femtoseconds means 10 to the minus 15 seconds, okay? A particular laser technique which is responsible for obtaining short pulses is called mode locking. It's not possible for me here to give a lecture on laser physics, so it's sufficient to say that this is the technique to produce ultra-short laser pulses. It was discovered by chance by Gurs in 1964 when he was doing modulation experiment for optical communication. He did not understand the result he got, and then 1 year later, my old friend Di Domenico explained the phenomenon observed in terms of locking of the modes. But at that time, the work was done with helium neon laser, so the pulse duration was not really strikingly short, namely 0.3 nanosecond, or if you wish, 300 picoseconds. The real breakthrough for as our interest came in about a couple of years later when using mode locking, but in solid-state lasers, like in ruby or neodymium glass, which were the only two solid-state lasers surviving, these people, in particular these two people, were able to go down to about 10 picoseconds. And this opened the field of using these picosecond laser pulses to interrogate the nature of these very short pulses. In that period, a very known and very important contribution from our group was to obtain a mode-locked ruby laser oscillating on a pure transverse mode. This was a key element for our success, which allowed us to obtain 5 picoseconds, which was appreciably shorter of what everybody else was doing. But in particular, since it was working as a laser should do, I mean with a pure TM00 mode, then the laser was quite reliable and reproducible. And this allowed us to do a lot of pioneering experiments in the 70s for nonlinear optic effect like self-phase modulation in nonpolar liquids for several years. Now I come to a new kind of laser which has been popular for at least 25 years, in which we have also given some contribution, namely dye lasers. They were independently invented by Peter Sorokin at IBM and Fritz Schaefer here in what is it? Hanover, Göttingen, in Göttingen. It is a quite interesting laser because it has got very large oscillation bandwidth, and furthermore, since you can easily change from one dye to another, these dye are used in liquid, in solution, you could easily cover with this dye laser the whole range from the near UV to the beginning of the infrared. But this laser became very popular in terms of mode locking again, because due to the large bandwidth, people, and in particular I should like to mention the pioneering contribution by Eric Ippen and Chok Shank at Bell Labs, were able to break the picosecond barrier, because at that time solid-state lasers were not able to get pulses below a picosecond, and to get down to a femtosecond. They were getting pulses of the order of between 27 to 100 femtoseconds. But the real solution why we could get such short pulses is to understand that the limitation to obtain short pulses was due to what is known as the cavity group delay dispersion. And we gave a lot of paper, we presented a lot of paper to prove that this was the limiting factor, so we had to compensate the group delay dispersion if we wanted to have the shorter pulses. In particular, the dispersion of the cavity mirror. There have been a few papers on that, quite a few. And then I come to another laser which is nowadays very popular, namely titanium sapphire, which was independently invented by Moulton and Huber. Moulton in the United States, Huber here in Hamburg. And it's very important because it has got a wide tunability, very wide tunability, 400 nanometers, which is 10 times the one of dye lasers. And but again, once you have wide tunability, you can get short pulses. using this titanium sapphire laser and performing model locking, Wilson Sibet and Michel Pichet in 1991, they were able to obtain pulses down model locking by a technique known as KLM, which is not the airway company, but it stands for Kerlens model locking. And they were able to get down to a few femtoseconds. And this finished the era of dye laser. This established the death of dye laser because dye laser were complicated to handle because they were liquids. You had to use pumps to circulate the liquids and so on. So people using laser, dye laser in those days, perhaps they could have some problem like this. You could have these liquids, which are colorful, being spilled over your suits. And that was not particularly pleasant, of course. And so, ending up this first part of my talk about these ultra-short laser pulses, this is a picture, a graph of how the pulse duration with different lasers have been shortened down through the years. I'm talking between 1966 to year 2000, so it's almost 40 years, 45 years. Solid-state laser, you see, the pulse duration has not been changing that much in this 35 period up to 1990, because the laser were only ruby or neodymium glass, and better than what we could get down to 3-4 picosecond was not possible. Meanwhile, when the problem of dispersion was understood and overcome, the pulse duration in dye laser rapidly or very rapidly decreased down to about 30 femtoseconds. But then, at the beginning of 1990, solid-state lasers, tunable solid-state lasers, were introduced, pumped by semiconductor laser or other lasers. And then at that point, the pulse duration of solid-state laser immediately decreased from a few picoseconds to a few femtoseconds, 4 or 5 femtoseconds. And this is the reason why the dye became obsolete. What can you do with ultra-short laser pulses? There have been 30 years of development and using ultra-short laser pulses to study the nature. I cannot review, of course, 30 years in 3 minutes or a few minutes. I like to mention time-resolved spectroscopy of molecules or semiconductor, the dynamics of molecule or semiconductor with high resolution. And here I again like to mention these two pioneers in the field, Ippen and Shang. Femtosecond spectroscopy of transition states during a chemical reaction, what is later on been named femtochemistry. And for this development, the pioneering development, Ahmed Zeewill actually, got the Nobel Prize for chemistry in 1999. And it was a big event because Ahmed Zeewill is Egyptian. It was the first Nobel Prize being given to an Egyptian, so they issued special stamps. There was a big celebration. And, of course, the other topics which have been covered through the years is the time-resolved spectroscopy of molecules of interest for biology, biomolecules, like rhodopsin, chlorophyll, porphyrins, and so on. In this regard, I like to mention an important contribution from our group on rhodopsin. A few preliminary about the physiology of the eye. You have the eye here, the retina. Light comes in from the crystalline, is focused on the retina. In the retina, you have the cones and the rods. Within the cones and the rod, there is a transmembrane protein, which is called rhodopsin. Within this rhodopsin, which is this is the membrane of the cellule, transmembrane of the cones or rods, you have the retinal. The retinal is a molecule like this, and this retinal is the molecule which is really responsible of the mechanism of vision, in the sense that when the retinal absorbs a photon, then it undergoes a kind of what is called in chemistry isomerization, means it is transformed by this configuration, which is named 11-cis retinal, to this straight configuration, which is named all trans. This very simple movement of this molecule then produces a lot of changes in the rhodopsin and then in the cones and in the optical nerves, which then allow us to see. And the mechanism of vision is quite efficient because we are almost counting photons. And the efficiency is so strong, is strong this way, just because when the light is absorbed, immediately after absorption, the light is transformed to other states, so it is not degraded in other ways. The secret is fast. Must be very, very fast, the transformation from this configuration to this, to avoid other dissipative channel to be operative. So it was very important to understand how fast this operation occurs and in what way. Pioneering contribution in this field, for instance, were given by Chok Shank, but a recent important contribution has been given by our group, in which we observe the movement between 11-cis to all trans with a fantastic resolution of 10 femtosecond, following every 10 femtosecond what is happening to the molecules, to the retina. So we absorb light of the retinal, go to the excited state, and then immediately we can follow through special techniques which are called pump probe technique. We observe that the packet of excitation moves here and reaches this position in 50 femtoseconds, very, very short time. And then it moves here to this and then goes to the other state, which is the all trans. So all this phenomenon is completed in 200 femtoseconds, and this was already studied by Shank. But all this detailed happening has been elucidated by our group, and this was published in the prestigious journal Nature. By femtosecond laser writing, we have been able to write complicated structure like this. I don't have time to discuss too much because the time is running fast. But essentially, we have been able to demonstrate a phenomenon which in the literature is called Anderson localization, predicted by Anderson quite a few years ago. Anderson predicted that if a coherent particle, if a quantum particle, enters in a perfectly diffusing media, then by coherent scattering through all diffusing particles, there will be interference of these scattering processes so that the light of the particle will preferentially concentrate in certain points. And this is named as Anderson localization of the particle. It's not diffused uniformly due to the wave nature of the particle. This is the Anderson localization by which Anderson got the Nobel Prize 10 years later. We have been reproducing this phenomenon of Anderson localization by femtosecond written waveguide, by focusing femtosecond pulses on a glass. We can write waveguides even in three-dimensional structure, and we proved that in this case we were able to obtain localization of entangled photon, but it's different story and different concept. Now, I go to a concept of compression, another big topic in our field, in which you can compress pulses by phase modulating the pulses and then compress in appropriate delay line. That's a very well-known scheme which was used also in radar physics. The phenomena which is responsible for this is self-phase modulation, but I don't have time to discuss about that. The only point I like to mention that through self-phase modulation, you can generate a lot of frequency modulation so that the band of your laser can be enlarged quite a lot. So that if you enter, for instance, in an optical fiber through here, the beam entering here is infrared. You don't see it. But as soon as it gets in optical fiber, this phenomenon of surface modulation generates so large band, that the beam becomes white, because if you have all the combination of visible components, then you have a white beam. So at the end, you have a white beam, which if you send the beam through a grating, diffraction grating, you see that this beam contains all components from the blue to the red. This is surface modulation. And another big idea, but I'm sure that Gerard Mourou is going to talk about that, is church pulse amplification, by which you can increase tremendously the energy of these pulses. But I'm sure that Gerard will talk a little bit about church pulse amplification. And here we come to another important contribution from our group, namely to use, instead of optical fiber to generate this big bandwidth and then the possibility to compress these pulses, we use a hollow waveguide filled with noble gases. And in this way, we were able to compress pulses of a relatively much higher energy. With fiber, you could not enter into a fiber with energy larger than 10 nanojoule, otherwise you won't have a fiber anymore. While in this case, we could modulate and therefore compress pulses up to a few millijoules, so increasing the energy of our pulse, which could be compressed by 6 orders of magnitude. And this has been a tremendous improvement in our field, this idea of the hollow fiber, which has been published in this paper. And by this technique, by the way, in 1997, together with a group of Fer and Kraus, we were able to get what was the world record at the time of 4.5 femtoseconds. Of course, we are not been hunting in getting records, because record value, after all, are the destiny is that they will be overcome sooner or later. But just I mentioned this just to prove that we've been in the forefront of the research for nearby all these 50 years. And so by compression, we could get down around the year 2000, down to about a few femtoseconds. But of course, we won't go down again to shorter pulses. The technique to go down is high-order harmonic generation. Here again, I cannot go deeply into that. But if you now send a very short pulses into a gas jet of noble gases, if the intensity is strong enough, then you can generate harmonic of the fundamental up to the 100 harmonic. If you generate the 100 harmonic, this means that the wavelength is reduced by a factor 100. So if you start with 800 nanometer and you divide by 100, you get 8 nanometer, which is soft X-ray. So in this way, we can generate short pulses in the X-ray regime. But in particular, it's important if we get very short pulses, as we can do with our hollow fiber. Because if you use very short pulses, as you can do with hollow fiber, then this harmonic in X-ray can come in only one pulse of attosecond duration. Attosecond now is 10 to the minus 18 seconds. So it's 1,000 times smaller. And so using this technique and using the hollow fiber, thanks to this technique of harmonic generation and use of hollow fiber, we have been able to get down in the middle of 2000, down to 180, 130. And so you see, this history is repeating itself. We started with dye laser, and we noticed that it's a logarithmic scale. So we started with a few picoseconds, we got down to 30, 40 femtoseconds, and then with this titanium sapphire, we got down to 3, 4 femtoseconds, and now we are going down, and I learned yesterday that the present record is not 130 anymore, it's 67 by another group. So, but why do we need attosecond pulses? Attosecond pulses very likely will establish new frontier for physics because it will allow you to study the motion of electrons in atoms or molecules, or the motion, new frontier for chemistry, the motion, electron motion for chemical reaction, or even perhaps new frontier for electronics, like electron motion in semiconductor nanostructure or molecular system. So please note that in all these cases, the important message that now, for the first time, we are able to study the movement of electron rather than we did before the movement of conformation of the molecule, which means the movement of atoms, which is completely different. Atoms are much slower than electrons. And this is now a modern laboratory with myself, and I'm not here to notice whether the passage of these 50 years of my face, if you compare the two, here I am in the face of the oscilloscope, or the richness of the laboratory. And by the way, at that time, we had only one laboratory and 5 people, 3 researchers and 2 technicians. And now we have 30 laboratories of this sort at the department, physics department of Politecnico. Now, I don't want to end up my talk without mentioning some early development, historically speaking, on laser application, early development, many applications which were tried failed. It was a disaster because the laser was not working well, the laser wavelength was not well. And this is the reason why in the early days, the laser was named a bright solution looking for a problem. That was the reason for the title of my talk. And so the final comment on our research activity. I hope I have shown you a coherent evolution of our activity over a 50 years period. It has been a strong evolution also because from initial 5, we have reached about 50 people now. From initial 1 lab to 3 labs, 30 labs. We have an institute of the CNR, an European large-scale facility, and the research we are doing are still promising, very exciting. So in this regard, we don't see the end of the story now after 50 years. And in more general term, laser early days, as I told you, was a bright solution looking for a problem. Laser 50 years afterwards, the bright solution for many applications in science and technology. 21 scientists, including the last 2 being given last December to Arosh and Weiland, 21 scientists have been awarded the Nobel Prize for research done on laser or with lasers. It has changed the way we live by internet, DVD, disc, and so on. That is in the letter by Barack Obama for defense. It is one of the most important inventions of last century. It's certainly going to play a very important, even more important role in this century. And that's why many people name this century as the century of the photon. Thank you for your attention.